Well, where we're headed today is we're going to begin this idea of the physiology of excitable cells. We're going to try to now superimpose a certain set of electrical parameters onto biological membranes. And we'll start here by laying out something known as the potassium battery. Potassium battery is simply a means of separating charge across a phospholipid bilayer. And this idea of separating charge across the phospholipid bilayer really lies at the heart of all the electrical activity inside all cells, but it's particularly true of neurons and muscle cells. We need to have this potassium battery in order to store charge, and then use the energy stored in the battery to then propagate and provide for electrical signaling in cells. You remember we said a major idea in sensory transduction was the change in movement of ions across the membrane. Well, this is the first place where we're now beginning to look at the details of how those electrical gradients are set up and how they're used as a part of transduction. And we'll talk a little bit about that. The first thing we're going to do, though, is do a kind of magical model where we're going to think about how we could possibly separate charge across a membrane and generate from that a sort of battery. Now, when we do this, we're going to find that we're going to use three major characters. There'll be potassium ions, large negative anions, and potassium leak channels. And we're going to do this as a kind of thought experiment so we can walk through this to think about how conceptually we can separate the ions, and by separating the ions now get a charge across the membrane. Then what we'll do is we'll take one step farther and we'll look at this in terms of a little bit of a mathematical equation. We'll talk about the Nernst potential, and I put this on the board the last time. Now, Dr. Nernst was, in fact, a physical chemist in Germany in the early part of the 20th century, and he derived the Nernst potential from some of the Boltzmann equations, which were some of the early stuff, the early work in quantum mechanics. But the Nernst potential here is going to simply be a ratio of potassium ion concentration on the outside of the inside. So we'll establish it first conceptually, and then we'll try to put some numbers on it so we can see how this is done. And we'll do this in a simplistic way, and then we'll talk a little bit about the biology. But it's not enough simply to generate a potassium battery. To get proper signaling across the membrane, we have to invoke other channels as well. And the first channel we're going to invoke here is a sodium selective channel. So now the first time we're going to put out potassium channels, and they'll be open all the time. They're going to be the leak channels. And then we're going to invoke a set of new channels called sodium selective channels. We're going to see that by opening these sodium channels, we can change the polarity on the membrane. We can go from minus 70 or 80 millivolts here up to plus 40 to plus 60 here, simply by opening the channels. And we'll see conceptually how that gets done. Now, one of the ideas that will fall out of this, and fall out of the Nernst potential, and fall out of the idea that each of the ions has an equilibrium potential, is that this concept of ion permeability and the idea that the membrane potential seeks the equilibrium potential for the ion whose permeability is dominant. Now, that sounds like a mouthful, but it's a good mantra to hold on to, because it says that if the dominant permeability is to potassium, which we see at rest, the membrane potential is going to be close to the equilibrium potential for potassium. If, on the other hand, we start opening sodium channels, then the predominant permeability will be to sodium. And now the membrane potential will seek the equilibrium potential for sodium. Now this begins to show a switch, where we can now show a change in the membrane potential from about minus 70 or 80 millivolts up to plus 60 or 40, and then back down again. And we'll see how this can happen. That is, by changing the permeability to certain ions, we can now switch the polarity across the membrane. That idea of switching the permeability from potassium to sodium back to potassium again, that idea lies at the heart of the action potential. The action potential is the long-distance signaling pathway that we talked about the other day when we looked at the functional description of a neuron, and we talked about the conductal region. It's the conductal region through which, along the membrane of which, action potentials will propagate. So what we're going to do first is we're going to set up this action potential. We're going to think about how we can turn it on and turn it off. Then we're going to think about this idea of threshold. Turns out that an important idea of how action potentials work, very important idea about how the potentials can propagate long distances up to a meter or more, lies at the heart of that lies this idea of threshold. That once a certain number of the sodium channels open, they provide such a large amount of positive charge that all of their neighbors open. This then leads to the sequential opening of the channels that we can now get propagation. For this system to work, though, the sodium channels remain open for only a short time. If the sodium channels remained open for a prolonged period of time, the membrane potential would hang out at the equilibrium potential for sodium, because after all, the membrane potential seeks the equilibrium potential for the ion whose permeability is dominant. And so if it's in fact sodium, then you'd be up around the sodium equilibrium potential. So we have to think about how we manage to inactivate those sodium channels. An important idea. Turning them on, turning them off, and then preparing them again for another round. Up to this point, all of this movement of ions can be thought of as a movement across the membrane in place. These potentials are not going to go very far. Then what we'll do is we'll talk about the idea of propagating a potential. How do we manage to move a potential from place to place? How do we manage to get these potentials to move along those conductor regions of the axon? And we'll do this in 3 steps to try to illustrate the importance of these voltage-gated channels. We'll first talk about a set of membranes, an imaginary set of membranes, where there are no channels. We'll only put in voltage-gated channels at one point, and we'll see those potentials propagate for short distances. And then we'll put them in unmyelinated axons and myelinated axons we talked about last time, trying to bring that full circle.